Welcome to TYT Interviews. We've got a fascinating one today for you guys. There's actually 3 different doctors we're going to talk to about stem cell research. We're here in YouTube Space, New York. Let me introduce the doctors first. Dr. Todd Evans, Dr. Jim Chung, and Dr. Albano Melli.
That's right.
Okay, as good as I can get it. Okay, so now this is a fascinating story. It involves a couple of different countries and one person's heart, and how some of these doctors have track the mutations and hopefully fix some of the issues that are at hand. It involves stem cell research. It's enormously fascinating. The more I talk to them off air, the more I got a sense of how much I love science. Okay, so in my mind, these are the good guys. And so let's have an interesting conversation about how it all works and how we all came to be in the same room. So Dr. Chung, let me start with you. We have someone on staff, Praveen Singh. She's the head of business development for the Young Turks. And she had an issue with her heart, and she came to you. So what was her issue?
So what had happened was that Praveen, several years before seeing me, had actually had an event where she just completely blacked out while at her office on the phone. It was actually quite a significant event. She wound up getting hospitalized. She was found to have an arrhythmia, had several procedures done, and had actually done well on treatment. But as it turned out, she had found out that her mother actually had the same condition. So actually, then fast forward a few years later, that's when she met me in my office. She had explained to me, and she was actually really an advocate for herself, because she really, she was actually pregnant at the time and said, look, I really think I have a genetic problem here. I have, at the time, a diagnosis of what was called short coupled torsade de pointes. It's quite a mouthful, but what it is, it's a form of arrhythmia that tends to affect patients who have otherwise normal hearts. Okay, so totally, she was young, otherwise healthy, no other medical problems, but she passed out from an arrhythmia. So it turned out that her mother had the same exact condition, so 2 very rare occurrences, and she had the wherewithal to think, this could be genetic. What can we do to maybe find out if there's an actual mutation that we can identify, with actually very direct implications for herself, because she was pregnant at the time for children, with a child, and she was worried that is it possible that I might pass this along to my child and future generations. So that's really how this all started.
So let me understand that, because that's pretty scary for a lot of people. So you might not realize you have this condition.
That's right.
And you might simply have, is it cardiac arrest that you go
Right,
into?
pretty much.
Right. And do we know if it's the same thing or not necessarily the same, but similar to the conditions we see on TV from time to time? Like if you're a sports fan, Reggie Lewis died suddenly while playing basketball. And do we know how many people it affects? Is it similar, and how overall, how much does this affect the country?
Right. So thankfully, it's very unusual. So a lot of patients who have cardiac arrest tend to have had prior heart problems, like patients who've had myocardial infarctions, meaning they had a blockage to a part of their heart where part of the heart has died as a result. Or a patient has a very weakened heart, what we call cardiomyopathy. So that, to just put it in perspective, is still the vast majority of what causes cardiac arrest. However, there is a distinct subset of patients who have otherwise totally normal hearts, who are young and otherwise healthy, who may have some unbeknownst condition that predisposes them to have cardiac arrest. So exactly like the examples you've mentioned. So Reggie Lewis had something called hypertrophic cardiomyopathy, where he had a thickening of his heart. Obviously, it led him to have electrical abnormalities of the heart, but he was a NBA basketball player, so obviously it wasn't affecting him so much that would prevent him from competing at a very high level. So every so often, absolutely, we hear these stories of marathon runners or other athletes who are otherwise very much high performing people who may drop, pass out suddenly, or even have a life-threatening arrhythmia. So again, put it in perspective, it's relatively infrequent. So for example, just to put in perspective, the most common form of this kind of a disorder called long QT syndrome affects, 1 in 1,000, 1 in 2,000, let's say, which is not it's not super rare, but it's definitely out there, but it's not, super common. But it's definitely something we need to know about.
So about 280,000 people a year die from heart attacks, but it's a smaller subset that are affected by this. I think what scares a lot of people is that it just seems to come out of the blue and there's no other indication. We all know to get our cholesterol checked. We all know to do our checkups, and so you have a sense of how you can make your heart healthier. But if you have this, boom, you're either, in Reggie Lewis's case, you're exercising and all of a sudden you get it, or in Praveen's case, you're at rest, and that's even scarier in some way. You didn't even know that was possible. So now, talking about that case, in her particular case, they know what it is, you guys know what it is, and you know that it's a mutation, right? So we think it might be genetic, but we don't know, right? So now, how do we find out more about it? How do we then transition to Dr. Evans?
Sure, yes. So basically, so with Praveen's urging, actually, we decided to send off genetic testing. Actually, to be honest with you, I was quite skeptical in the beginning because I felt that her story did not fit. So there are a list of disorders that cause sudden, cardiac arrest in otherwise healthy hearts, and there's a list. And her condition did not really fall under the known list, which has been known to be associated with specific mutations. So obviously, there are a lot of genetic abnormalities for which are out there that we just don't know what genes they are because we haven't explored every possible gene out there. So at the time, I actually was a bit skeptical about doing genetic testing because I felt that, what's the likelihood of us finding something is going to be low. And actually, there are downsides to finding something because if you find something, that mutation, the alteration of genetic code, may or may not be what's actually causing the condition. So what happens is that the downside of doing genetic testing when you don't know what you're testing is that you may found some abnormality which is actually totally benign, it's just as benign as difference in hair color, but then you link it to some potentially, you think, fatal condition without the full scientific knowledge of what's going on. So in any case, just to backtrack, so in Praveen's case, we did the genetic testing and we found a mutation in a gene, which was really quite surprising because that mutation is associated with arrhythmias generally with exercise. And in her condition, it occurred purely at rest. So at the time, we was like, well, that's interesting. And then we tested the mother, and lo and behold, the mother had the exact same mutation. Now, that doesn't prove anything, right? So that's the thing is, now we have two patients with relatively uncommon presentation with a relatively rare mutation. And then now, however, the hard work begins, which is making sure we can prove that we have the science to make the association between the actual mutation and the actual medical condition we're trying to make the diagnosis for. And that's where, Dr. Melli and Dr. Evans steps in here, where we can actually give us the scientific evidence to support that hypothesis.
So already this is really interesting and a little scary for people if they have this, and so they're rooting for you, I think, to get this right. But I think it's about to get more interesting to see what Dr. Evans and Dr. Melli have done. So you send Praveen's blood to Dr. Evans. Dr. Evans, what do you do with the blood?
Well, what our group did with the blood was we're actually turning them into stem cells. What we'd like to do is be able to study Praveen's cardiomyocytes or other cellular components of our heart, but of course we can't do that. A physician can study various clinical aspects of her condition, but we'd actually like to understand the molecular basis for what's going on, to actually be able to understand the biochemistry in terms of how the proteins are functioning in hearts. And there are certain things we can do now that even 5 or 6 years ago we just couldn't do. A lot of times, for example, you could study somebody's blood cells and try to make an assumption about how a gene being dysfunction there relates to a cardiomyocyte, but that's a big stretch. What we'd actually like to be able to do is generate cardiomyocytes, sort of replicate, do what we call the disease in a dish, actually create functioning cardiac tissue in the dish, which has the exact same genetic makeup of the patient. And we can do that now. So what we do is we can take blood cells and introduce 4 genes that are called pluripotency genes. They're genes that are important for imparting a phenotype of a stem cell, similar to an embryonic stem cell, that allows these cells now to become reprogrammed so that they forget basically that they're a blood cell. Actually, one of the beauties of this process is that blood is easy to obtain just from the periphery. Everybody goes to the clinic and gives a blood draw. We need very little blood. We use the blood cells that will normally make red blood cells, which is the most abundant cell in your body, and so we can expand in a petri dish these what are called erythroblasts, and we introduce a virus, which is an engineered virus, and that virus is such that it will go into these red blood cell precursors and express these pluripotency genes, essentially erasing the memory of that cell as a red cell. And the culture conditions are such that we can capture cells as they transition and reprogram and become a stem cell, and themselves become pluripotent. The reason that's important is that one of the great features of a pluripotent cell is that we can grow them in a dish, and we can make as many of them as we want. We can grow them up, grow them up, grow them up. And so that's occurring right now, actually, at Weill Cornell Medical School in our incubators. Now, the beauty is that over the last 5 or 10 years, that's something that my own group is very much involved in, but many other laboratories are working in actually using these embryonic, these are actually called induced pluripotent stem cells, iPS cells. It doesn't do us much good just to grow up lots and lots of them, but we have to do something with them. And so a lot of people have been working on the protocols to then differentiate them into different cell types. So you could start with a blood cell, reprogram it back to the stem cell, make a lot of it, and now induce it to turn into whatever cell type you like. And one of those, which we now have very good protocols for, are cardiomyocytes, or cells that make the beating heart. We're also very interested in making other cells of the heart, like the cells of the conduction system that's involved in controlling this rhythmic beating, and we're very actively involved in that as well. So we can make those cells in the dish. And as Dr. Chung was saying, it's just absolutely true. Even if you took a cell and, for example, put in that mutation, even in a cardiomyocyte, it's not really what we want to study, because the genetic background will be different and genes interact with each other. So what we really need to do is have the exact genetic background to understand how that affects cardiomyocyte function.
Okay, so let me try to break this down. And I love this because a lot of times I deal with politics, and you guys are so much smarter than the politicians that it's exhilarating to talk to you guys. Okay, and I'm trying to learn as best as I can. So now you can turn the red blood cells into stem cells, and so already that's amazing.
It is amazing. So we were chatting earlier, that was one of the pioneering events that did lead to the Nobel Prize in 2012 by Shinya Yamanaka and his colleagues.
So when you got the stem cells, can you, if I'm understanding it right, and that's where I need to help here, you can make them into heart cells of that particular person, right? Can you do other cells, liver, kidney, you can do all of that. And so then you can begin to study how that person's heart cells reacts when you introduce a disease into it, to be able to track it. Is that roughly right?
That's roughly right. Now, there are many caveats to this because, of course, even though you can make, for example, heart cells, cardiomyocytes that really are from the patient, essentially, I mean, secondarily derived, but they're not a heart. And so they're not affiliated with the whole physiological response that occurs in a living human being. So it's a very much of a surrogate assay. That's what we call it, disease in a dish. But they do function. They'll beat. They'll have hopefully the same physical characteristics as they did in the person. You can now have as many of them as you want, and so you can look for what other proteins or genes might be altered in those cells. They're very easy now to add drugs to test, to screen for whether or not drugs give certain responses. So it's a way to bootstrap actually from a patient into essentially a clinical trial in a dish to think about pharmacological approaches to working on the disease.
So now I'm going to make it personal for me. I have a fairly rare skin disease. Okay, it's called pemphigus vulgaris. I don't know if you guys are familiar with it. No reason you would be, right? It's not in your fields. So could we then take my kids' red blood cells and test them? Is that a different course of action, or to see if they would have it also, because it is genetic, and then how we can treat it afterwards? So my point being, forget about me. forget about Praveen, can this be applied across the board with other genetic diseases?
Very widely, and there's many scientists now actively engaged in that type of research, whether it's, for example, neurodegenerative disorders, diabetes, complex genetic diseases. Cystic fibrosis is a very well understood disease, but one of the things we have been working on recently is generating, for example, pancreatic ductal cells that are the target of this disease, a major target of this disease. And there are many mutations that can cause cystic fibrosis, but now you have a screening platform to actually look for drugs for, not just for cystic fibrosis, but for the cystic fibrosis that that person has because of not only the mutation in the known disease gene, but all the other genes in their genome that interact with that. And we need, and it's really, this is what has now been called precision medicine or really medicine that's designed for a patient, not for a disease.
Okay, now we're not done yet. It's about to get it more interesting. Okay, now we take the heart cells that Dr. Evans has made and we ship them over to France to Dr. Millie. And what do you do with them?
So basically, based on the clinical features that Dr. Cenk can reveal, we focus on some and based also on the mutation occurring in some particular protein, what we call in this particular case endin receptor. We focus on all the calcium that is released into the cardiomyocyte. So calcium is a major component to allow the contraction, actually precede the contraction. And what we do, so back to France, we focus on all the properties that are related to the calcium into the cells. And for that, we basically use different techniques. And also based on what we did with Dr. Cenk so far, we know which part of the cells and which protein in particular we should really focus and try eventually to model the disease. So from basically the initial patient through the IPS via Dr. Evans, we get cardiomyocyte and we try to model some features of the disease in the dish, still in the dish. Okay. So then what we can do once we have those features, those phenotype, we basically can try different drugs that are either under clinical trial, so FDA approved, and see which drug would basically prevent or treat any of those features that we can reveal.
So again, let me try to see if I've got this right. Correct me where I'm wrong. Red blood cells, then stem cells, then heart cells. Okay, and then once you've got that person's particular heart cell, you then introduce the diseases into it, and then do you test it against a control to see what the difference is?
Yeah, well basically the idea of patient-specific cardiomyocyte is that the mutation is there already, okay?
Mm-hmm.
And thanks to Dr. Evans' expertise, we actually can correct some of the stem cells to get a perfect control. So the only difference between the two cell type is just one mutation.
Okay, so let me pause there because the audience might not quite understand that because I had trouble understanding it in the beginning. So normally you would find a person who's about the same age, who's also female around Praveen's age, and that would be your control group to see how that reacts differently as opposed to the one with the mutation in it, which was Praveen cells. But you guys don't do that anymore. Dr. Evans actually fixes the mutation so that it's Praveen's heart cells, but that are fixed, and that's your control. And then you test it versus the one that has the mutations.
Absolutely. Yep.
Phenomenal.
So all the rest of the
Phenomenal.
genetics is the same.
Yeah. All right. So then you introduce the disease and I'm sorry, you have the one that's mutated, and then you what do? You experiment with how to fix it? I mean, it's in layman's terms.
We try to exhibit some of the features of the disease in the dish, and only in with the cardiomyocyte. Try to reveal some of the features, either by applying some pharmacological compounds that will activate or inhibit some proteins, or we try also to improve the maturity of the cells by some physiological or pathophysiological stimuli, just to reveal the disease. And once we have those features, once we characterize the cells and can reveal the features, we then go to the second step, which is basically testing different drugs and see which one would be the best for the patient in the dish, and maybe eventually for at the clinical level.
So, Dr. Chung, would that then come back to you and say, okay, Dr. Millie says this is the one that's most likely to work for Praveen, so then when we eventually try it and see how it works for Praveen? Is that roughly right?
Right. I think that, thankfully Praveen's doing well right now. So, there's no, she's doing okay with her current therapy based on the previous procedures she's had. So she's doing quite well, but it has obviously implications for other patients like her. So, so again, with Dr. Mellie's work, that will first at least establish that the mutation is indeed disease causing. That's the first thing. That's what we need to do, is we need to prove that it really is, this mutation really is causing a problem here. And if he can recreate the disease in a dish, then that ups the evidence level for the fact that, okay, this mutation is causing a problem. And then if on top of it, like Dr. Mellie says, if he can find the best compound to treat it, then that will be, again, even more, with even greater significance. And then it comes back to me, and then we can think about things, then we can think more big picture. Okay, so how many people are out there with this disease that we don't know about who may have the same mutation? Then you may talk about employing ideas of screening, patients. Now, it's a relatively rare disease, so this is something where it would require multiple, across the nation and international type effort to identify patients with her condition, and then doing more widespread screening. And if we find that, hey, a certain percentage of patients, usually never 100%, but let's say a good substantial percentage of patients with her similar condition have this mutation, then that would, or at least of the same gene is affected, then you can think about using that same drug that tested so well in the Petri dish in a, say, clinical trial.
And if you already had a parent that had these kind of conditions, well, then you're far more likely to know that you should test. Right. And so what it might do is, for the people who had been dealing with this and it's a mystery and they don't know why and they have to live in panic the rest of their lives, am I going to, literally fall down one day and not get up and have this heart attack in a way that couldn't have been predicted? Well, now we can test for it. And then, so one more part of this that I have to understand, you guys build this whole system for understanding it, for mapping it, etc. Then when a person comes in, is the medicine personalized for them? Is there a process you build where you take the blood and it becomes easier, so that you guys are building the beginning of it, but it's got to be easier than going through all three of you for each patient, right? How do we make that process a little more efficient?
I mean, the hope is that down the road, that this will become more of a widespread approach. But right now, this is just one case, We're just fighting the battle on one particular patient who, again, had the wherewithal to say, hey, test me. There are tons of people out there that we don't have this, either don't have the resources, we don't have the technology, we don't know which gene. So the thing is, we need to take a step back. We don't want to whole scale start screening everyone who has a genetic condition for something, because then you pick up random things that we don't know whether they're of significance or not. So, but down the road, as we sort of start cataloging more of the genes that are associated with these genetic disorders, then we can sort of do more of this approach. But it's still in its infant stages, but obviously, it's hopefully something, as Dr. Evans had pointed out, this idea of personalized medicine, which, that President Obama had raised in the State of the Union address. It's sort of kind of that idea.
So, Dr. Evans, is it that then once you've mapped out all of this, that still you take somebody's blood from them, and then you bring it through this process and you go, well, the medicine most likely to work, given the research we've done in the past, is this? Is that how it...
Well, I would say that, of course, there's two aspects. One is the screening that we're doing now is this physiological screening in a dish to try to replicate the disease. You're not going to want to screen people like that generally. So, but if for example, this mutation is identified as really being responsible for the patient's disease, then it's easy to screen for that genetically. You would just add it on as part of standard genetic testing, ultimately, if it was really a strong indication of the disease. So genetic screening, there are standard genetic screens right now for more common diseases. We all imagine that at some point it will become standard protocol to have your genome sequenced. That's controversial because there may be, first of all, there's a lot of issues about protecting patient privacy, and that information is a patient's own or a newborn's own genetic information. But ultimately, I think we all believe that at some level that will become standard of care, and but it still is very complicated because of the fact that, as we brought up several times, even the identification of this allele. I mean, I can pretty much guarantee you, and it probably was part of the reason why there was some skepticism, and we're still very, not skeptical, but we're going to test a hypothesis here. We don't know if it's correct. That same allele, which is called a mutation, a difference, a gene difference, in a different person might not have any consequence at all because they have other protective genes, for example.
Okay, and so Dr. Melly, you're trying to find out what different medicines could work to help if you already have this condition. But you were saying earlier that Dr. Evans fixed a mutation in order for you to have the control test on it, right? So can we fix the mutation in the body without treating it with the medicine?
Well, that's a very good question, and there's a lot of fantasy around such a possibility. I'm sure maybe in the future it might be possible. Currently, this is not the case, and for many reasons. But like you just said, indeed, what we try to do is to get the perfect control. The perfect control is just a single point mutation between the two that makes a difference between the two samples. Because if you pick one patient and another, you always have a genetic background that differs, and some genes might be more expressed in one side or the other side, and so on. Question in the future, can we just fix a mutation like this in the whole patient, in the whole body? Technically, it might be possible in the future, and Dr. Evans is maybe more expert than I am to answer this. But I think currently, definitely, this is not possible.
So right now it's enormously complicated because of what you were just saying. There could be different things that are reacting within each person. So even if you figure out that one mutation, and you try to fix that, it might affect other things in your body. Is that right?
Well, there's two issues. One there's that, but I think more importantly right now, I mean, technically it's feasible to do. It certainly has been done in animal models. In mice, single mutations have been corrected and cured the mouse. Now, currently, the recommendation and from coming through the NIH for federal funding is that we shouldn't even be doing these experiments in humans because it's just too early. We need to take the time to think it through, because if you can correct mutations, you can do any kind of editing that you want in the genome, and it has serious ethical consequences. So currently, at least in the US, we're not doing any types of experiments like that in people, other than for use in a dish. To actually study the biology behind specific changes in the genome. Okay,
but somebody could.
Somebody could.
Right, so even if we're not doing it here, somewhere in Outer Mongolia.
Well, there has been a report from a Chinese group that they have modified the human genome. And of course, doing it in a manner where there would be no opportunity for embryos to survive or emerge or actually grow. So done at very, very early stages of embryogenesis. And there was a bit of outrage about that, in that I think generally scientists worldwide believe that this is very powerful technology, and it's okay. We have so much to do. There's so much we don't understand. There's so much great experiments that can be done that we can do those for a while as we work through this and the ethical issues and where and how the human genome should be modified.
Okay, so now we're going to get a tiny bit into fantasy world here, because what the work you guys are doing is super real, it's very important, and it has real consequences for people in the real world. Okay, but now that we've opened up this interesting, I don't want to say Pandora's box, but so I do want to blow people's minds a little bit. If you, quote unquote, fix a genome within a person, not treat it with medicine, but fix it within the person, if they have kids later, they would actually pass on the new genome that you corrected. correct?
It depends on how you corrected it. So you have to correct it not in, you could correct it in every cell in the body. If you didn't correct it in the germline, so the cells that will give rise to either sperm or oocytes, then you're not going to pass it on. So it's very possible, you could imagine in science fiction world, coming up with strategies that fix the gene in every cell in your body or in just some cells of your body. So what we would call tissue-specific correction. Say, let's fix the gene in all the cells of the heart, because in fact that's the only place that this gene being mutated we think is important. Other genes maybe are important in all cells, but if it's done in with a strategy where you wouldn't modify the germline, then it wouldn't be passed on.
Mm-hmm. And could you, if you were so disposed, change the genome in a way where the person would be taller?
That shouldn't be probably difficult. You could
Stronger?
Stronger.
Uh-oh.
Absolutely.
Okay.
So these are the so-called designer genes that you know, there are certain physical characteristics that are fairly straightforward to alter, knowing what people know about growth and development and hormonal systems, etc. So
when would they, so one last piece of fantasy here and then we'll get back to the real world. When would they do that quote unquote fix? Let's say you wanted to. It's 38 years out. It's not here. It's in outer Mongolia. Somebody says, I want to make a super fast, tall, strong person, right? And I want to do it for whatever reason. Would you do it when it's a fetus, when the baby is born? Can you do it to an adult?
Well, we're in super fantasy land here. So it's always going to be easier to fix something with current technology the earlier you do it, because it's just there's less cells to fix. So if you could fix genetically edit at the one cell stage, or what's called a zygote, so a fertilized egg, that would be by far the easiest place to do the editing. And then it would fix every cell in the body. Now, of course, that would only work if you're starting from one cell. If you're starting from a grown adult, then it's much more challenging. But for example, it should be very feasible right now if you had there's a way that you can mix stem cell biology with gene therapy, which is, there are trials ongoing right now that are basically adding genes, for example, to your hematopoietic stem cells that make all the blood cells of your body. We can take those out of a person's body, genetically alter by adding a gene, and those stem cells can be put back in, go back to the bone marrow, repopulate your entire body, and so you can cure diseases that way. And there are clinical trials to cure blood diseases like thalassemia, for example. Now, it should not be difficult. In fact, today it would be feasible to use the same approach, but instead of adding a gene, actually go in and fix the mutation in that sense, and then all the blood cells would be fixed, and it wouldn't be passed on. So those kinds of therapies, probably in the next 10, 15 years, may be moving all the way to clinical use.
Okay, so that's a perfect way to come back to today and the work you guys are doing. So it has both very specific purpose. So for example, Dr. Chung, if it turns out, knock on wood, one of Praveen's daughters has the same mutation as she does, well then all this work can hopefully find medicine to be able to more effectively treat her if she needs it, correct?
Yes.
And so that's very feasible fairly soon. 5-year window or so, roughly speaking. Okay, so that's great that we might be saving people's lives. Your work saves people's lives today if this comes to fruition. So that's phenomenal. And then secondly, there are broader implications, because that's just one example. And then you can take this and multiply it out, as Dr. Evans was just talking about, to other diseases and see the cases in which you can specifically design medicine or perhaps even fix it within the body. Is that right?
I think so, yeah.
That's phenomenal. I have to say, throughout the interview, I kept thinking in my head, because I'm a goofy guy, science. All right, well, I speak for the entire Young Turks community and our audience in saying thank you. We really appreciate the work that you do on behalf of not just the people that we know, but humanity. So thank you, Dr. Evans, Dr. Chung, Dr. Melly. It's been a great pleasure talking to you.